So beginning with this, we all understand how biology is immensely complex and that we are always searching for new tools to understand fundamental biological processes. Traditional tools like qPCR, flow cytometry, bulk RNA sequencing that most of us are very familiar with have allowed researchers to get us this far in our standing. But as we move forward into the next era of science, we need better and higher resolution technologies to take the research to the next level. And so today we are going to learn more about single-cell gene expression sequencing, which is the newest tool that is allowing to resolve complex biological problems better than ever before. So before we delve into this, I'd like to take a look at some of these traditional technologies that have brought us to where we are today and how single-cell sequencing actually will allow us to take those next steps into the future. So I want to begin at looking at RT-qPCR. It is a bulk method which allows us to quantify the gene expression level of specific genes of interest using pre-designed gene-specific primers. With RT-PCR, we start by extracting the bulk RNA from our sample and then using reverse transcription to convert the RNA into cDNA. And once we have that cDNA, we can then quantify the expression levels of our favorite genes of interest using our pre-designed primers. So qPCR really is a great technique because it is cost-effective. It's a cost-effective way of looking at a few genes of interest. However, it has some limitations as well in that it is a bulk method. So we do lose the heterogeneity of our sample. And additionally, it is really only feasible to do this for a few selected genes at a time. And lastly, those genes you actually have to know about. So you really are not able to discover new biomarkers. So the ability to use qPCR as a discovery tool, it is not there. The next tool, microarrays, they are also a bulk method, but microarrays provide the additional benefit of now being able to profile thousands of genes at a time. These tools are glass slides that are printed with thousands of tiny spots in very defined position, and with each spot containing a known DNA sequence or gene. So they act as probes to detect gene expression from the sample with a fluorescence-based readout. So similarly to qPCR, it is a bulk method, but it is able to profile many more genes compared to the qPCR method. However, you still have to know which genes you're interested in profiling in order for this to be useful. So while they are a great validation tool, they are also less suited as a discovery research tool. And then traditional RNA sequencing or bulk RNA sequencing is also a bulk method, but it is able to profile the whole transcriptome of a sample rather than just a few specific genes. So like qPCR and microarrays do this with just a few genes. It does this by first creating cDNA libraries from RNA based on the poly-A tail of the mRNAs. And then we are adding primers to the library that can then be used for next generation sequencing. And then from there, the whole transcriptome can be profiled, which really allows the discovery of new important genes. However, this method also is a bulk method. So the complete profile or the complete heterogeneity of the sample is lost. So when we look at bulk RNA sequencing, really we can compare this sort of with a fruit smoothie. Here we make certain assumptions of the cell population. We can include or decide on the composition of the sample based on some sort of very prominent markers, but the full picture really is much harder to understand and to put together due to the lack of the fine resolution. So single-cell sequencing really differs here because it is like looking at the bowl of fruit straight on. You get a much more detailed, complete profile of a sample without the need of guesswork and assumptions. So now that we have an understanding of sort of the benefits of single-cell RNA and how that compares to our traditional methods, I really want to look at what the data gets us. What does single-cell data get us? So as we mentioned, biology is highly complex, and most sample types and systems that we study are complex and highly heterogeneous. So when we take these gene expression profiles of a bulk sample with a bulk method, we are essentially taking just an average of that heterogeneity and averaging it out and measuring it. So we are losing parts of the information. So if we were to profile the same sample using single-cell sequencing, we can see now that we would be able to look at each subpopulation of the cells and understand which cell types is contributing and is expressing which gene. This gives us a much clearer picture now and an understanding of the sample's heterogeneity. So when we look at, what strides we have taken with single-cell sequencing, let's take a look at what single-cell data looks like. Here we can see an illustration of a t-SNE plot, which is one way that scientists can use to look at and interpret single-cell data. And the t-SNEs are a method to reduce complexity of data sets into a two-dimensional way to visualize the representation that we can then more easily understand. So in this illustration here, each dot represents an individual cell or a nucleus, and cells that are closer together, plotted together in this t-SNE plot, have more similar transcriptional profiles than cells that are further apart from each other. So these cells can then be grouped together into clusters. So we've indicated this here by the different colors, and that is done based on their transcriptional profiles and using gene expression markers that are characteristic for specific known cell types. You can then go ahead and actually annotate these cell clusters. Now on the right, we are showing an example of a real-life t-SNE plot. In this example, you see obviously a lot more cells profiled than in our illustrated version. And in this experiment here, the authors profiled thousands of cells from IBD patients, inflammatory bowel disease patients, so bowel tissue samples, and you can see the different cell types that the authors were able to discern from the sample. So single-cell sequencing has advanced extremely far in a relatively short period of time. So if we look at the beginning of single-cell, if we look back sort of 10, almost 20 years ago, sort of the first publications on single-cell contain just a few cells, usually under 100 cells. And they were typically conducted by researchers developing these methods in their own labs, so homebrew methods. They were very challenging and were usually only able to be executed by very well-versed researchers in this field, and often limited to qPCR analysis to actually allow the profiling of the expression differences. Now, moving forward the last 3 years, the picture looks very different. We now see a large number of publications where many more, up to 1 million cells are profiled and reflected in these publications. And that is due to mainly the fact that we now have commercialized solutions available to perform single-cell RNA sequencing, and it does make it a lot easier for laboratories to adopt this technology and start their own research. And at 10x, we have just recently passed the 5,000 publication mark. We are really excited by this, and it does showcase the impact researchers are making in their fields using the 10x products.